Now, after this structural comparison, let's have a look at their functional comparison. We know that both BCRs and TCRs are antigen recognition molecules that are known for their vital role in adaptive immune system. But here let's see how their function differs from each other. Although their primary function is antigen recognition, but the mechanism for both varies. Let's see how, starting with B-cell receptors. Well, BCRs or B-cell receptors have two antigen binding sites. And we know that these sites are made of both light and heavy chains. And these light and heavy chains are actually the protein or polypeptide chains. And proteins are made of what? Amino acids, of course. So, these sites are responsible for antigen binding because they exhibit tremendous diversity in amino acid sequences. So, what I want to convey is that the antigen binding site of the BCR, formed by the variable regions of the heavy and light chains, contains specific amino acid sequences that can recognize and bind complementary antigenic epitopes. Now, what does it mean? Well, we know that antigens are the substances that can elicit an immune response. These antigens can be large molecules such as proteins, carbohydrates, lipids, or nucleic acids. However, the immune system does not recognize the entire antigen as a whole. Instead, it focuses on specific regions within the antigen. And those specific regions are named antigenic epitopes. These epitopes are the small, accessible portion of the antigen molecules. And they are typically composed of a few amino acids in the case of protein antigens, or specific sugar residues in the case of carbohydrate antigens. And then the specific molecule, such as B-cell receptors, binds with that specific portion on antigens, and thus easily recognize them. So, let's look at this recognition mechanism, followed by BCRs. We know that the antigen binding site of BCRs is formed by the variable regions. And these variable regions consist of hypervariable loops called CDRs, or complementary determining regions. These regions are made of diverse amino acid sequences. And so, these CDRs directly interact with the antigenic epitope, determining the specificity and affinity of the BCR antigen interaction. This binding between BCR's antigen binding site with that of epitope can be compared to a lock and key mechanism. The antigenic epitope acts as the key that fits into the lock formed by the CDRs of the BCR. And as particular key fits into the particular lock, similarly particular antigen binds with the particular CDR. The shape, charge, and chemical composition of the antigenic epitope determine its compatibility with the BCR's antigen binding site. And here, several types of molecular interactions contribute to this binding, such as hydrogen bonding, electrostatic interactions, or van der Waals forces, etc. The greater these forces, the greater will be the binding affinity, that is actually strength of binding between BCR and antigen. And as we said, this affinity is extremely specific. And this specificity and the affinity is the result of diversity of BCR that is generated through genetic rearrangement that we will discuss later. So, by any of these forces, binding of an antigenic epitope to a BCR can induce conformational changes in the BCR, triggering intracellular signaling pathways that lead to B-cell activation and subsequent immune responses. These signaling events are crucial for the B-cell to initiate processes such as proliferation, differentiation into antibody-secreting plasma cells, and the generation of memory B cells. So, this was all about the functions of BCRs. Now, let's compare it with that of T cell receptors. Well, we know. Just like BCRs, the primary role of TCRs is the recognition of antigens. But this recognition mechanism differs from that of BCRs in many terms. Like, TCRs do not recognize antigens directly, except for gamma delta TCRs, but their amount inside the body is very negligible. So, here we will talk in reference to alpha-beta receptors. So we can say, majority of TCRs do not recognize antigens directly. But they need a specific intermediate molecule that is called antigen-MHC complex. And as name suggests, this complex is made by the union of an antigenic peptide plus an MHC molecule. We have detailed videos on this specific personalized equipment of the cells. Here I am talking about MHC. And you can unlock the mysteries of this major histocompatibility complex by watching our many videos on this interesting topic under the titles MHC and Antigen Presentation. What is major histocompatibility complex, MHC, class 1? And what is major histocompatibility complex, MHC, class 2? Coming back to our topic. We were saying that TCRs require the complex of antigen with that of MHC molecules to recognize and bind the specific antigen. And there are two major types of MHC molecules. That are MHC class 1 molecules and MHC class 2 molecules. The location, type of antigen binding, and interaction with particular subset of T cell all varies for both these types of MHC molecules. Such as MHC1 molecules are found on almost all nucleated cells. But MHC2 molecules are found on antigen-presenting cells only. MHC1 molecules interact with the endogenous substances. But MHC2 molecules interact with exogenous antigens or substances. And lastly, MHC1 molecules present antigen to cytotoxic T cells. But MHC2 molecules present them to the helper T cells. Let's have a brief look at the interactions of TCRs with that of particular class of MHC. Starting with TCR-MHC class 1 interaction. In this interaction, the T cells involved are cytotoxic T cells, also known as CD8+ T cells. These cells express TCRs that interact with antigens presented by MHC class 1 molecules. And the antigen presented by MHC1 are the endogenous antigens, which are derived from intracellular proteins synthesized by infected or cancerous cells. So, once this endogenous antigen is processed inside the cell and interact with the binding site of the MHC molecules, this complex gets anchored over the surface of that respective cell. That could be infected or cancerous cell. Now imagine the wandering CTLs or cytotoxic lymphocytes with the T cell receptors or TCRs at their surface. That one comes in close proximity with the antigen MHC class 1 complex, they trigger and binds to that complex, allowing the T cell to recognize that there is some problem with that cell. So, these cytotoxic T cells that are known for their killer activities take necessary actions and eliminate the infected or abnormal cells from the body. So, that's how TCR interacts with MHC class 1 molecules. Now, let's look at TCR-MHC class 2 interaction. Now, here the subset of T cells involved is helper T cells, which are also known as CD4+ T cells. These cells express TCRs that interact with antigens presented by MHC class 2 molecules. And the antigen presented by class II molecules are the exogenous antigen. And these exogenous antigens are derived from extracellular pathogens that have been engulfed and processed by APCs. Until the particular size amino acids are formed. These amino acids then make bond with the binding site of MHC class II molecules within the APCs. And then this complex is anchored over the surface of antigen-presenting cells. And when this antigen MHC2 complex encounters the TCRs found on helper T cells, they initiate a signaling cascade, leading to the activation of the helper T cell and the initiation of immune responses, such as the activation of B cells and further cytotoxic T cells. So, that's how TCRs interact with MHC class II molecules. Now, if we summarize the function of BCRs, we say that antigenic epitopes bind with BCRs through specific interactions between the epitope and the variable regions of the BCR. This binding is compared to lock and key mechanism, and the lock and key binding involves molecular interactions such as hydrogen bonds, electrostatic interactions, and van der Waals forces. The affinity and the specificity of these interactions are crucial for the effectiveness of the immune response. But once it's done, it activates a signaling cascade inside the B cells and decide a specific fate for the antigen and the cells. On the other hand, the TCRs require a specific complex for their binding with the antigens. That complex could be antigen MHC class I complex and antigen MHC class II complex, where in first case endogenous antigens are presented by MHC class I molecules over the surface of infected or cancerous cells. Similarly, in the second case, exogenous antigens are presented by MHC class II molecules over the surface of antigen-presenting cells. Both these markers are actually informing the immune cells that there is some problem that needs to be checked. So, once these abnormal protein fragments are presented over the cell surface by MHC molecules, then this presentation acts as an alarm bell, summoning a special force of immune cells called cytotoxic T lymphocytes, CTLs, or killer T cells, and helper T cells respectively. And these remarkable defenders possess receptors capable of recognizing the unique combinations presented by MHC molecules. And then they take necessary actions against the presented antigen to help our immune system and to protect the delicate balance of our existence. So, this was a comparison between the functions of BCRs and TCRs.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.